Perfect. So my name is Ashley England. I'm a swine field specialist for IPIC. I work out of the northwest corner of Iowa. I'm housed in Sioux County. Today, we are going to have Dr. Andersen talking to us about foaming manure, safety, science, and solutions. So this is a hot topic recently, and we love to get Dr. Andersen's insight on it.
All right. Thanks, Ashley, and thanks for having me today. There we go. All right. And if you were one of the three people who called me about foam so far this week, I'm sorry I didn't get you called back. I'm hanging out over in Denmark for the week looking at anaerobic digesters. So I promise I will listen to your voicemails and return your call if you're one of them. All right. So they asked me to talk about foam safety. We've had a few reports, somewhere in the neighborhood of 14 over the last few weeks to month in Iowa of people who've had a flash fire. And I know there's been some other calls that I've gotten from around the nation, so definitely something that we're struggling with again. So I just wanted to talk through sort of what we know, what we don't know, and some safety precautions that we can take to go with it as I try and screen share work. There we go. So to begin with, I'm just to talk about what we look at when we see foam. I'll go through some of the dangers and some of the safety considerations we had. And then we did a fair amount of research on this back in 2014 through 2016. And while some of the maybe causes have changed slightly, I think what we found then is going to be really applicable to probably what we're seeing now. So we'll walk you through sort of what the science said and how that relates to some mitigation strategies. All right, so just a couple pictures of foam to get us started. We sampled a lot of foam, and I think we learned a few things about it. There's a few things that really stand out to me when I look at it, and I'm showing you sort of two really extreme pictures here. That bucket on the left is really frothy. It's still filled with methane bubbles. If you take that foam, bring it back and let it sit around long enough that those bubbles start to pop and you don't make any more inside it, you get that gooey sort of substance there that looks like either really bad meringue or really bad whipped cream. But I think it really gives you the picture of what the stuff around the bubbles looks like, right? It's sticky, it's gooey, it's filled with solids, it's really viscous. And then when I look at that foam, there's a few things that really stand out to me. The color is one of them, right? We really look gray to dark brown. And that sort of helps set a diagnostic tool of what kind of foam it is, right? So when we see that sort of color, it often implies that there's protein in that foam. So just as we're eyeballing foam, if you see something that's frothy and white, that's normally related to sort of short chain fatty acids or surface tension, and where you might think in our head, that looks like a soap bubble. If you see sort of that gray brown color, like you might get on, well, a head of beer, sometimes the beers turn a little whiter because there's alcohol in them. That's generally a protein-derived foam. All right, so the dangerous part, right? And I think this is where we really want to start. In general, methane is lighter than air. So as manure is making methane, we do have this small layer on top of the manure surface that's higher in methane concentration. But since it's lighter than air, it diffuses up into the barn, and our ventilation system normally helps get it out of the barn. So when we look at that picture on the top, that's sort of what I'm depicting for you there, right? There is some methane down there. It might even be at high concentrations right above that manure surface. But as long as our barn ventilation system is running, functioning the way it should, we're generally getting it out of the barn at the rate we're making it. And there's not a lot of safety concern. The exception to that would be when a barn is empty and we turn that ventilation system down, or hopefully not off, but I know I've visited some barns in Iowa when there's no pigs in there and they want to save some propane and they shut it up. If we aren't getting that methane out, we can run into some concentrations in the barn that get more dangerous levels. But generally where we tend to have the largest cause of concern is in barns where we've had this foam buildup on the surface. And that's really because the foam traps the bubbles. And if you do anything that breaks the foam, agitate, start soaking your barn with sprinklers or washing your barn right out, as that water, that material falls through the slats, we break those bubbles and it releases large amounts of gas relatively quickly. And methane is explosive at concentrations between 5 and 15%. So those foam bubbles below the pits, when we think about methane generation from manures, we normally think somewhere in the neighborhood of it's 50 to 60% methane. Well, the truth is in the foam, it's a little higher than that. It's about 70%. And that's got a lot to do with carbon dioxide dynamics and how it dissolves into the manure water around the bubbles, but a large amount of foam there. So if we break those bubbles, we tend to be in that explosive 5 to 15% range. So we have to get that methane out of the barn. Okay, so a few things to think about when you have foam. So this time of year, hopefully we don't have to worry about headspace quite as much, but if we start getting late summer and we have foam, the first thing you're always thinking is it going to get into my pigs? What sort of headspace am I working with? And is there a field I can get some out, right? Because getting some headspace created so that when we do break the bubbles and release those gases, which is going to be methane, hydrogen sulfide, we don't want it right in the pig breathing zone. So the first thing I'm always thinking is there something I can do to help with that headspace situation? After we've done that, I'm really thinking about what's my foam management situation here. Am I just trying to get by for a couple of weeks until I'm really going to pump out? Is this a long term? I have to make it till my fall pump out if it's something that's happening right now. So one thing you can always do is sprinkle with water. If you're going to do that, I write on there use caution because you're releasing a lot of methane relatively quickly. So it's something we have to do. We have to get rid of those bubbles, but we want to be ventilating our barn, minimizing the potential for sparks when we do that. I also wrote add anti-foaming agents, and we'll talk a little bit more about what I mean by that later in this presentation, but I'm generally talking about surfactants. So that could be a silicone-based surfactant. It could be other things. I know some people have used crop oils in the past. Crop oils, I generally think we're trying to buy ourselves 2 weeks, mostly because crop oils break down with microbial activity. We tend to have plenty of microbial activity in the pit. So they won't last forever, right? And as they break down, they tend to make some of those foaming situations worse. If we think more about purchased surfactants like you might be using in spraying situations or those silicone type surfactants, the microbes in manure don't break them down nearly as quickly, right? So that can be a longer term fix. We can also think about microbial controls. And I wrote sort of the easy ones there. We could think a pH shift, so adjusting the pH of our manure or adding oxygen in our manure. And I know both those aren't very practical, but I'm sort of trying to set the perspective of if we can actually change the microbial community, we get some foam control. So the treatment that most people start with, and the one that I recommend first, is some sort of antimicrobial, usually an ionophore, Rumensin, Narasin, which is Skyces, or Coban, tend to work relatively well. 5 pounds per 100,000 gallons of manure. And really what they're trying to do is disrupt that process chain of making methane. You can think about other ways to try and shift the microbial community, and pit additives, especially microbial additives, are potential ways to do that. Some of them may work more effectively than others. I obviously haven't tested all of them that are out there. One of the things that we did test was bacillus, and bacillus break down protein. And I told you that based on the color of the foam, we often are looking at protein-based foams. No matter what you're doing, if you're in this situation, the biggest thing we have to pay attention to is what are our sources of sparks, right? So when we think about the fire triangle, we need three things: a fuel, well, in our case, that's methane. We need oxygen for the fire. There's plenty in the air, although if the foam, that methane is captured in the foam, for the most part, if we first start on fire, it's oxygen starved, but plenty of oxygen in the barn. And the last thing we need is an ignition source, so that's spark. So the big things that I think we need to be paying attention to: cigarettes, we shouldn't be smoking in the barn, especially if we have that foam situation in there, sparking or arcing switches or motors, and then probably most importantly, welding and grinding. Hot work in the barn should always be done with caution. I would really recommend slag mats so nothing's falling down in the pit. But especially in foaming situations, that's where we've seen most of the issues, right? It's people who still think, I need to weld, I need to fix this. If possible, get it out of the barn. If it's not possible to get out of the barn, we really have to be thinking about slag mats so that nothing falls in. All right, so even on the previous slide, I talked a little bit about treatments, but I want to reiterate a couple things. One, if we are doing anything to break that foam, we are going to release methane, and the methane is going to get to explosive levels in the barn. We have to get it out. So we're really doing everything we can to minimize ignition sources during that time period, and then ventilate it at a minimum of 30 cubic feet per minute per pig space in that barn. Get people out of the barn and treat your pit, right? So whatever the treatment might be, but we really have to get rid of the sparking, arcing potential at that time and have our ventilation system up so we're trying to exhaust the methane as we go. Steve Hoff did some of the work on what it would take to dilute out methane right through our ventilation system, and thankfully it is heavier than air. What he generally said is we need 6 minutes of time. So of course, Iowa State and I say you need something like 15 to 20 minutes to get all that air ventilated out. So even after you've done your treatment, keep going, make sure that we get back to background levels. So 20 minutes of ventilation at that higher level before you go back to your minimum ventilation. Okay, a little bit on foaming theory. So it really takes three things to make a foam. The first one is we need some bubble source, and for us, that's going to be biogas. And I don't think that comes as a surprise for anyone. There's not too many sources of what that bubbles can be. I know as our manures get a little foamy or have more foaming issues, you might see them pop up when we're adding manure to a tank, right? And that foam's not explosive, but it still is annoying because we don't get our tanks as full. But for the most part, if it's just sitting there, we're getting foam on it, it's a biogas situation. The biogas is really methane, carbon dioxide, and hydrogen sulfide with really small amounts of ammonia. The two that I'm most concerned about, really, like we talked about, that methane for the explosion potential, and hydrogen sulfide. The hydrogen sulfide, we normally think about it as release when we agitate manure, and that's really the dangerous time period. Here it's that we have trapped some in that biogas bubble. And if we break the bubbles and it's right in the animal breathing zone, there's potential for issues. The other thing is surfactants. And when we talk about surfactants, I generally think about blowing bubbles in water versus blowing bubbles in chocolate milk, right? And one's definitely way more fun than the other. You want to think your chocolate milk. So what's the difference between the two? And it's that there's surfactants. There's stuff in that milk that help lower the surface tension and make it easier for bubbles to form. So generally in milk, we're thinking about either the proteins helping with that or with some of the fat in the milk helping with that, right? So there's different types of surfactants and they all make a difference. And then the final thing that you need is really a stabilizer, right? And that's just something that holds the foam bubbles together and makes it stick around. So if the bubbles form and they pop instantly, sort of like you can do with water, it wouldn't be a problem. So we need to think about what's the potential surfactants and is there anything we can do about it? All right, I wanted to feel like a scientist for just a second. So back when we were doing this work, I think there was really two things that people were talking about. They said, well, it's something new microbe. Maybe we have this brand new microbe that's causing this foaming issue. And we'd say things like selective pressures can cause big differences, and that's why it's occurring. We'll talk to some of the science about that. The other one that often came up is, well, we're feeding a lot more DDGs today than we used to. It's a major ration change. That's the only thing we feel like we changed. It must be causing foam. I know these days we see more foaming issues even on barns that aren't feeding DDGs, and I'll sort of talk through why that might be the case for you. But I tried to put those into maybe a little bit more of a sciencey term or what we really wanted to test or understand as we were doing this work. And if you think about it was DDGs, that's really saying we think it's higher fiber diets that are leading to more carbon in the manure and microbes eat that carbon, right? So that would be a potential mechanism. Following up on that, you might say, well, if there's more carbon there, are they turning it into methane at a greater rate, right? So that was our second hypothesis. The third one is really small particles maybe are present now that stabilize the foam. What could they be, and can we figure out what they are? And then the last one I said was, it's kind of like an emulsion that they're making. So let's think of foam like meringue, and does that help us understand anything? I did get a question in the chat and I lost it, but it was a question about sky seas, and I want to stop and address that one for just a second. So I'm going to show you a slide later about us dosing sky seas directly into the manure and how effective that was for me. When you think about feeding it, it has the same general effect. About half of its activity is lost in the animal though. So when you feed it, you're trying to get to my high rate of what I included manure, but since it's half broke down, you get to sort of my middle rate. So it should still be effective for us. I think maybe the struggle when you're feeding sky seas is we generally aren't feeding it for every phase of the ration. So when I give you that dosing rate of, I'm trying to get to 5 pounds per 100,000 gallons of manure, if you're only feeding it for a few steps in the ration, you won't get nearly as big. Now, if you fed that sky seas when your pit was full and we put it on in the manure early, it probably changes the microbial population that developed. If the pit was mostly full and we end up with nursery pigs where we're feeding the sky seas in the ration, it probably doesn't have as much effect on what's going to happen, right? So it can work in our feeding trials where we fed it. It definitely caused the manure to have less foaming characteristics. I won't guarantee that's always true. I've definitely seen people who feed some sky seas who still have foam, but they probably aren't getting to the dosing rates that I'm recommending that we try and get to in manure. All right, I wanted to talk just a little bit about studying the biogas phase. So there's just some pictures of what we were doing. There's lots of ways that people have done this before. And really, we modified one of those methods. So one of the things, since I'm on this digester tour, that people often run is called a biochemical methane potential test. You put some inoculum in a bottle and you put your substrate, generally for me that's manure, in your bottle, mix them together, and then you measure how much biogas you make. And that's really good at understanding sort of the potential of a material to make methane. But in this case, I think we cared a lot more about is it really making methane quick right now or not? So we didn't inoculate the manure. We really wanted to focus on, is there a microbial community right now that can process carbon really effectively? And so I put them on my lab bench, just straight manure, let them incubate for 3 days at room temperature, and then came back and measured how much methane they made. One of the things that we saw really early on in this test is some of the bottles made foam on top, and that was really cool. You can think about what the foam was, why it was there, but more importantly, it gave a really big visual observation right away that that foam was present on bottles that were making methane more quickly. And you can also look at that bubbly mass there and sort of say, well, it looks like a few things are happening. I see some stuff separating, and it looks like more solids separate into that gunk on top, that floating scum, than stay in the manure, right? So I think there was things that beyond just the methane that you could look at and take away from this. With that said, I want to talk about the methane. A big thing to me was that barns that are foaming make methane more quickly than barns that don't foam by about 3 times as much. And that's not a lot of gas or air moving through the manure, but it's a significant difference. So in the wastewater treatment world, one of the ways people try and separate certain types of solid is with something called dissolved air flotation. So you put micro bubbles down at the bottom as they move through whatever you're trying to separate solids from. Some of those solid particles stick to the bubbles. They ride the bubbles to the surface and you get this mat on top and you get less solids in the manure. So methane's important here because it's making bubbles, it's filling that foam up on top with bubbles. But I think it's also important for us because it's working as a separation mechanism, right? So it's both filling those bubbles up, and that's part of the cause, but it's also moving some stuff to the surface that would naturally normally be in the manure. And that separation of stuff is equally important in causing why manures have some of these foaming properties. And that's sort of what we'll talk through. But when we say treat with an ionophore, really that ionophore is focusing on how do I take something that's making more methane than we generally would think it would, down to a manure that doesn't make as much methane per unit time. So it's treating filling those bubbles, but it's also treating sort of that phase separation problem. Okay, this is a slide I don't often show when I talk about foam, but I wanted to talk about it today for a very specific reason. So what I'm showing on the y-axis here, the vertical axis, is the rate of methane production. And we talked about how higher rates of methane production generally lead to more opportunity for foam. On that x-axis or the horizontal axis, I'm showing total solids content. And to me, when I look at this graph, it sort of says that in order to have the biggest opportunity to foam, I want to be in a solids content of somewhere between 6 and 8, 9%. So if I don't want my manure to foam, I generally want to have lower solids content than that, or get a higher solids content than that. And that's sort of to say that when we look back at sort of that 2012, 2014 era, we tended to be on the low sides of the solid content. And as we were switching to wet dry feeders, as we were feeding more distillers, we tended to be moving up in solids content to sort of the, hey, we're in this range where we're optimizing how much solids is in there to generate methane. And that's generally what people who have digesters try and operate at, somewhere in that 8 to 12% range, because people say that's where you generally optimize gas production. These days, almost everyone is using wet dry feeders. I know we're feeding maybe some more soybean in the ration. That should be bringing our solids content down, but we've gotten better at water management. So even these days, we're probably still hanging out in that 7, 8% solids content, sort of in that optimum range to cause foam. So that's sort of me saying that, well, I think back in 2010, 2012, to get to that foaming era, DDG sort of made a difference because they were enriching our manure and solids. These days, probably not as important to have a specific feed ingredient in there that might be causing that foam. It's more related to we're doing a better job with water management. And that doesn't mean we shouldn't be doing water management, right? That to me is something like 2 cents a gallon to not have to call it away and land apply it. So water management helps us be better utilizers of our manure nutrients, but it does lead to this sort of, we're in this range where solids content might be pushing us towards manures that foam. All right, the next thing we tried to study, and I just wanted to show you the super scientific apparatus we built. It's just a PVC tube, nice and clear. We put an aquarium stone at the bottom. We put manure in, blow air through it for a short period of time, and see how many bubbles we could get and then how long those bubbles stuck around. And we didn't learn anything groundbreaking on this, right? But when you look at how much it would foam or its capacity to foam, we did tend to see that foaming manures foamed more, right? When you blew air through them, they'd make more bubbles. What I'm showing on that x-axis this time is just stratum. So B is right at the surface of the manure. C would be at my midpoint of manure, and D would be down in sort of a sludgy layer as close to the bottom as I could sample. But those foaming manures, especially near the surface, were accumulating stuff that gave it greater capacity to foam. No surprise, right? Like that's what one would expect, but at least it tells us that we were quantifying something that relates to what we saw. The bigger one to me was how long the foam would stick around. So we would wait days for the foam to break down, and we'd measure how long it took half of it to stick around. So in that sort of B stratum, you can see that there's a lot more stability to those bubbles. If I took the foam itself, blew air through it and caused it to foam, I got extremely long half-life, something like a day and a half. So those are sort of useful to understand, that we could see, quantify what was going on there. But they probably all make sense, right? Like I have foam, so those two things probably have to be true. And the real question is, what causes them? Okay. So when we talked about bubbles earlier, one of the things I pointed out was surfactants or surface tension. And when you think of those, I'm often thinking about something like soap bubbles, but it could be a surfactant that you add. And really the way they work is they have some region of the molecule that has a slight charge to it that we might call polar. It likes to be dissolved in water. And then it's got some long tail on it that's generally a fat oil. Something that doesn't like water is hydrophobic. So those things might encapsulate bubbles, right? They might have their polar head towards the water, their long tail inside the bubble, and that helps bubbles stick around, at least in theory. So we started measuring surface tension on a whole bunch of manure, partly because it's just so fun to do, but more so because we thought it would be a great diagnostic tool. And I found something that surprised me. And surprises are always good, right? The surface tension of foaming manure is actually higher than non-foaming manure. So originally we were hypothesizing that, well, it's making more of some surfactant that's sticking around and causing it to be more foamy. The truth is, non-foaming manures have more surfactant in it. It's generally short-chain fatty acids. So what's happening in that foaming manure is those short-chain fatty acids get consumed by the microbes that are making methane, and they're breaking it down, and they're bringing the surface tension slightly higher than it would be in the non-foaming manure. And one way to think about that, and maybe not the most politically correct way to think about it, but it's like a pair of underwear, right? So if you have an old pair of underwear and the elastics wore out, it won't spring back. Well, that's the non-foaming manure, right? So when you make the bubble, it basically pops instantly. Foaming manure is more like a good new pair of underwear, enough elasticity that as the bubble stretches and expands, it's strong enough to pull the membrane of the bubble back together and hold the foam in. So all that to say is, when we talk about why adding a surfactant might be helpful or might be useful, well, right there, right? We saw that non-foaming manures had more of that surfactant in it, lower surface tension. So adding a surfactant, I'm trying to do that. I'm trying to get to a point where those bubbles will break easily because there's not enough strength in that film to hold it together. Okay, viscosity. I said early on that foam is really viscous. And here I had tons of fun, right? So we're measuring viscosity. For perspective, the viscosity of water on this scale should be about 1. So anything above 1 is more viscous than water. A viscosity of about 30 would be a viscosity of something like motor oil, right? So it depends a little bit on the temperature, but 30 should be roughly motor oil. So one of the things we did is we just took that foam, the foaming manure and non-foaming manure, and directly measured viscosity on them. And there's no difference between the manures basically for any of the cases we did here, but the viscosity of that foam itself was much, much higher. So I think about that as, well, the water hangs around in the foam for a long time and it stays gunky or gooey or wet. And if it stays wet, the bubbles aren't going to break as well. One of the questions we had is, well, that foam is just really enriched in solids. Is this really a solids problem causing viscosity? So we centrifuged it to try and remove as many solids as we could. And we saw the same thing, right? The foam still had a higher viscosity. And I said, well, that's because I can still see solid particles in it. So we ran it through a filter to get every solid particle out of it we could. And we still saw the same thing, right? Higher viscosity than the manure itself. So that doesn't identify what the surfactant is, or what the cause of the viscosity is, but it definitely says there's something inherent in the liquid that could be causing it. It could be oil, it could be lipopolysaccharide, it could be protein, or if you have no idea, you just say a word like microbial goo and move on with your life. But luckily I made a friend. And that friend was Steve Tribune, and we started talking about this. And we did a few other things. One of them was throw a tannin into manure, just like you'd use to tan a hide. And I took a non-foaming manure, threw this tannin in, blew some air through it, and I made a nice stable foam. And I was like, Steve, what is this? We were thinking it was going to be a mitigation technique, and I was able to take this non-foaming manure and show that now I have potential to potentially make a foam. And Steve said, well, tannins interact with proteins. And all of a sudden, we stumbled on something that was actually semi-helpful in diagnosing what we thought was happening, right? Because we knew all of a sudden that something binding with proteins was super important for us. So I wanted to talk just a little bit about meringue. It is my favorite emulsion because it's super yummy on my lemon meringue pie, but it's also really applicable to what we're seeing here. So to make meringue, you take a protein, in that case egg white, and you start shearing it or whipping it up to entrain air in it with sugar. So the protein interacts with the sugar, and next thing you know, you get a nice fluffy meringue. All right, so let's try and relate this to manure. All right, so we knew that particles were important for foam stabilization, and the first thing we said was, let's measure the particle size. So upper left-hand corner here, I have a blue line, that's the particle size distribution in the foam. I have a red line, that's the particle distribution in the foaming manure, and the green is in the non-foaming manure. So when I look at that, what I see is particles between about 2 and 25 microns are super enriched in the foam, and those same particles are depleted in the foaming manure relative to the non-foaming manure. So that doesn't mean that the foaming manure isn't making those particles. It really is implying that those particles are riding the gas bubbles up to the surface and getting in the foam. So we're stealing from the manure, putting them in the foam. All right, so we learned something useful there. And then Steve did his magic and said, I can measure protein content and all these things. And what we found is that the foam itself is really enriched in protein. So all of a sudden we got lucky with throwing tannins in and said maybe proteins were involved. And here we were able to say, hey, we have evidence now that says proteins really are involved. Okay, so that's where we sort of learned that there was proteins as a big part of that. I'd like to tell you there was something magical about the protein. Most of the other work on it out of the University of Minnesota implied that it was probably corn protein. So probably not a whole lot we were going to do about that. We definitely feed corn to pigs. Distillers has some protein in it, but everything has proteins and it's going to be there to some level or some extent. All right, the next thing Steve did was he said, I think I can do better on that microbial goo. I could do a digestion for you and then measure total carbohydrates, hemicellulose, and fats and oils. So Steve started doing this digestion on the foam, on the foaming manure, and on the non-foaming manure. And what was really super interesting is I want you to look at this picture on the bottom. He did this digestion, then they'd centrifuge it. And in the foam itself, you can see that it got this black gooey schmutz on the top. If you digested either the foaming manure or the non-foaming manure, you did not get that material. So that material is sort of saying, hey, once you do this digestion, you can get a goo out of it that looks like an oil. To complement that, Steve ran total carbohydrates, so we could say, hey, the foam has more total carbohydrates in it, more hemicellulose in it, and that's a diagnostic tool to say there's some sort of lipopolysaccharide in there. So that microbial goo is a lipopolysaccharide, and we know it's interacting with the protein. So really handy for sort of understanding what the mechanism of foam is. Maybe not super helpful for understanding how to break it down. Okay, the last thing we did is the University of Illinois and then Adina Howe here at Iowa State did a fair amount of microbial analysis. So what we do is take a small sample, extract the DNA from it, and then they do a microbial fingerprint. And really, I think about that as like a microbial census, right? So they'd identify what types of microbes are present and then how much of the population they made. And that's too much data to really understand, but they would plot it on these two-dimensional graphs like I'm showing in the middle. And with those two-dimensional graphs, points that are close together are really similar in microbial population. Points that are far apart are really different. So if you see them grouping together and you can say, hey, foaming barns group together, that's an indication that certain microbial arrangements tend to lead towards foaming populations, especially if that's different than non-foaming manures. All right, so they did this. They made some graphs. You can see the open circle here would be a non-foaming manure. The orange circle would be a foaming, or the orange square, sorry, would be a foaming manure. And what you really can see is they cluster differently, right? So the stuff that's foaming, you can draw a circle around it and say, that's a community that leads to a foaming structure. Around the other one, you can draw a circle and say, maybe an oval and say, that's structures that lead towards these non-foaming characteristics. And then they overlaid some of the other things we were measuring on that with the length of the arrow showing how important it was or how related it was to have a foaming microbial community population. So the more the direction points to the center of that oval or the center of the thing we're interested in it, the stronger it was related to it. So the biggest thing that we found was that methane production rate was highly correlated with manures that were foaming, as was depth and the temperature of the manure. To me, all those things are related to the methane production rate, right? So if I have more manure, I have more gas flux moving through that manure because I just have more manure to make methane. Bigger chance of having bubbles, bigger chance of getting some of that stuff to separate. The other two characteristics that tended to be related to foam were either DDGs or fiber. So things that increase the methane production potential of the manure. So all that to say, most of this data to me mostly says that things that increase gas potential in the manure increase our risk of foam. So when I go back and say, hey, that solid slide was kind of interesting, right? That being in that solids range maximizes our methane potential. Well, that's what this data sort of aligns to, right? That things that increase solids content in the manure, things that lead to more food for the microbes to eat, make more methane and more chances for foam to form. All right, the other thing that they did was they measured microbial richness. And this is basically how many different types of microbes are in the manure. And we did this for two integrators. Big thanks to both of them for letting us collect bunches of samples from different barns in their sites over an extended period of time. And one of the things that we saw in both cases was that foaming manures tend to be less rich in different types of microbes. There's more of certain types of microbes that are doing things, and they are outcompeting others in there. So that's interesting. Since one of our initial hypotheses was maybe there's some new super microbe making foam, I wanted to show you that we didn't really see that. Certainly we saw that sometimes there'd be more of different types of microbes in foaming manure than non-foaming manure, but we also saw pretty much everything that was present in one was present in the other. All we were doing is changing some of the types that were out there. So in non-foaming manures, we did tend to see more lactobacillus. Bacillus bacteria are known to make proteases, things that break down protein. So some of the work we tried later was related to, does adding different types of bacillus bacteria help break down that protein, reduce chances of foam? In our foaming manure, we saw a lot of things. None of them really jumped out at us as this is the one that probably is causing that goo, right? But there isn't a lot of databases that help you understand all the functionalities of different microbes. One of the things that really did come through is that foaming manures are processing carbon and handing off their byproducts to different microbes better, so that we get from a piece of solids, piece of organic matter, all the way to methane probably more rapidly or more efficiently. All right, I promised a little discussion on Andersen. It was Andersen is SkySeas, so SkySeas is a trade name. Here I was adding it directly to manure. So I chose 3 different dosing rates. That middle dosing rate is 5 pounds per 100,000 gallons of manure. I just have it in silly metric units because that's what people like to publish in. So one of the things we saw is that it was actually relatively effective as soon as we got that product into the manure. If you are doing a treatment, it's important to get it into the manure. It's a powdery substance when you buy it. If you get it hung up in the foam and it doesn't actually make it into the manure, it is not effective. So I generally like to put it in each of the pump outs, add some water into a 5-gallon bucket and throw it in so it really makes it down through that foam all the way into the manure. Then it's a soluble compound. It will spread out and start doing its job. It was proportional in dosing rate to how much we added. 5 pounds per 100,000 gallons really probably is the ideal dosing rate. Throwing more in, I did get more methane reduction, but it didn't last any longer in how long I was getting methane production. So generally that lasted for about 80, 90 days as an effective product for me. Just as importantly, it changed the microbial community by adding it to our manure. One of the questions I think people often ask or I get when we talk about what we did is, does that kill the pit? No, it slows the rate we're making methane, but it barely changed the amount of solids I broke down between storage, between my treated and untreated manures. I changed the solids content by 0.1%. If I asked you to take two samples from your manure pit, excuse me, and they came back within 0.1%, I'd say, oh, you did a nice job sampling. Those are the same number. So it does slow microbial activity slightly, but it definitely doesn't kill your pit. So with that, I'm happy to try and address any questions that you might have or talk through some of the issues that you're seeing.
Thank you, Dr. Anderson. I think that gives us a lot more information about how and why, and definitely a better understanding of foam. Um, I guess one of the questions that I keep coming back to is as far as the safety aspect, if foam is present, what is maybe in a simple form, um, the safety that can be taken when they are ready to address the foam and treat it immediately?
Well, the first thing is making sure that if you're going to do anything to break the foam, we definitely have to be ventilating our barn. We should be running minimum ventilation in the winter, even when pigs aren't in our barn out of a safety precaution. But no matter what, if we're breaking that foam, ventilation is extremely important. The second thing would be uh, what are we doing to minimize chances of spark during that time period? And then my sort of bigger answer to that would be you should be watching your foam, checking for it. If there's more than 6 inches of foam, we should be thinking treatment. If there's less than 6 inches of foam, we're probably hanging out in the neighborhood of be observing it and tracking it to make sure it doesn't get out of hand. But we're probably not ready to treat until we get to sort of that 6 inches of foam area for me.
So would you say it's safe to say ventilate, um, get rid of any ignition sources, break bubbles, ventilate?
Yes, yes.
Okay.
Keep it on for 30 minutes afterwards. So ventilate to get that methane out of the barn is extremely important. I saw we got a question about diesel fuel. Uh,
Yes.
and surfactants. According to Iowa code, you are not allowed to add diesel fuel to manure. So that's sort of my answer on that. But surfactants can be an effective form of helping mitigate some of that foaming risk, right? And we showed some data on why that's true. Non-foaming manures tend to have lower surface tension, the bubbles break. So surfactants can be effective, but it is against Iowa code to be adding diesel fuel to your manure.
All right. And then the question after that says we've been seeing higher soybean meal use in the diet. I know we talked about that briefly. Could the higher dietary protein content be part of the reason?
Yeah, so that doesn't necessarily go with what we saw in the earlier study, but I think we've moved to more wet dry feeders. We tend to have the thicker manure, so I think we have that methane potential no matter what we're feeding these days, right? In terms of can the soybean meal be the protein that is binding in the foam? I don't actually know. I don't see any reason why it couldn't be, right? It just has to be able to interact with something, but we haven't really done anything specifically to study that. I saw we got a question now on will different surfactants kill the pit? That's a great question. We have very little information on what most of them would do in a pit, but I would presume that most of the ones that are used for surfactants in our agricultural situations will not. In aerobic conditions, they tend to break down relatively quickly. Even in anaerobic conditions, they probably won't break down as quickly, but most of them don't have a toxicity that's been reported to them according to the label testing they have to do. If you start picking random things to throw into the pit, I can't make any guarantee for you other than to say it's really generally pretty hard to kill a pit. You can do things to reduce microbial activity, but when you sort of look at the range of bacteria and life we have on Earth, manure pits aren't the greatest place to live. They're already pretty salty, and the bacteria find a way to live through that. They're pretty basic, and some scientific evidence has shown if we make them really acidic, like try and get that pH down to 3.5, we can kill a lot of the microbes in there. We won't kill everything. So you probably won't kill your pit, but you shouldn't just be saying, I have this laying around, I can throw it in, right? We should probably be thinking about what surfactants can we have access to that we use in agricultural situations, like spraying chemicals that we know won't cause environmental harm when we use this as a fertilizer.
Could we maybe take a step back and just define when you say kill the pit, what that would entail, what that would be?
Yeah, so to me, killing the pit would be something where we really slow breakdown of solids. So I think just hydrolysis of solids is a pretty good indication of what's there, right? So as long as we have some bacteria functioning in the manure, we're going to keep breaking down solids. Uh, so killing the pit would be really reducing the microbial activity to such a level that we stop hydrolysis of those solid particles and we're no longer sort of breaking them down. And that's actually pretty hard to do. I know some people who have used Andersen, Skyseas, Rumensin. Uh, the sort of the kill the pit theory, I think, comes from the fact that when we have a foaming manure, some of those solid particles are sticking to the gas bubbles and they're floating up, and it seems like it's not as sludgy. It's more uniform throughout the pit. When we don't have those gas bubbles, well, we get a better chance for some of those solids to settle. And it's not that you've actually changed breakdown of the solids in the manure, it's that we have a more liquid manure on the top and then a sludgy layer on the bottom.
So we've got a lot of questions coming in here. I'm going to back it up a little. If the methane gets trapped in the foam, is the methane level in the barn air very low? In other words, could you measure methane in the air and predict the likelihood of an explosion?
I mean, you could. You can buy gas meters that measure essentially explosiveness. But I think for the most part, the concentrations in the air in a barn under normal conditions, even when we have foam, are relatively low. The challenge with having a fire or an explosion really relates to those times when, well, we either have slag or something fall directly into the pit where we have that small layer of high methane, or we've done something to break the bubbles. So you could have a meter to help measure that. If you do anything to break the foam, you don't need a meter. We're in explosive concentrations, and you should be acting like we're in explosive concentrations. If there's foam present in the barn, uh, you could take a meter out and say, "What's my explosiveness of this manure?" And it will spit out if it's between 5 and 15%. Generally, we're not in a range where you're going to be explosive in those situations, whether you have foam or not. Like I
I
said,
know,
it's light
oh,
air,
go
so
ahead.
if our ventilation system is working, we will get it out of the barn.
I know you and I have talked in the past that when pigs are present in the barn, the bubbles are naturally being popped. There's always things
Mhm.
falling through the slats and the barn. The bubbles are naturally being popped. We're seeing the issue is when ventilation is down, barns are empty, and those bubbles aren't being popped, and then they're bumped. They're the way I understand it when you explain it is that they're all suddenly popped all at the same time.
Yeah,
Is where we have the issue.
that's right.
What effect does agitating the pit have when the barn is empty? Does that help with solids breakdown?
All right, that's sort of an off-topic question, but generally, yes, but it's a short-term help for solids breakdown. So one of the ways that you can get more microbial activity is getting sort of those solids suspended throughout the manure. But based on all the work we did for this, that effect lasts for like a month, and then you're back to where you started. So yeah, it could help you break down some solids, but long term, it won't cause a big change in your solids content because we're not going to agitate enough to do that.
I know you've been asked quite a few times, but do you have a specific pit additive brand that you recommend?
I don't, but I had my most luck with Bacillus bacteria. With that said, I think especially early on, if you were treating when the pit is empty, they are more susceptible to microbial change in community. So if you have a pit additive you like or are interested to try it, then would generally be the time. Empty the manure. We've caused basically a forest fire, right? And sort of reset the microbial ecology in there because it's a very different environment for a short period of time. So that's when pit additives would tend to be the most useful at causing change. I think many of them can do it, but there isn't a lot of work out there saying that this is really what we want our microbial community to be for solids breakdown or for odor control. One of the few things I can say is changing it from sort of that what was causing foaming to anything else basically reduces foaming, right? So I think a lot of products could be effective there. The ones that I've tended to try, I focused on Bacillus bacteria because we knew that proteins were involved and we know that Bacillus make a lot of proteases to help break down proteins. So I was really focused in that direction.
So another good question here. It says it's best to focus on prevention, but what is the best way to put out a pit fire if it occurs? Fire departments tend to want to spray water, which is not good and will release more methane. Is it best to ventilate and allow it to burn out?
Yeah, it's best to ventilate and allow to burn out. And that's easy to say and hard to do. If anything's on fire, our first reaction is, I want to spray water at it, right? Because it's natural. But like you said, breaking more bubbles right next to where the flame is will cause methane release and put us towards that. It's going to burn faster, be an explosive concentration. So you sort of have to ventilate and hope it burns through. Most of those have been flash fires, right? So they get over relatively quickly. I've talked to quite a few people who've been in the barn and said, I watched it burn sort of from one end to the other end, and then it was done. But it's not an easy thing. It's unnatural. You should try and get out, have some ventilation running, and then it has to run its course. Throwing water on top of it is going to break bubbles. And since we already have that fuel or ignition source, it's just going to release the methane as a fuel and we move towards an explosion.
So, another question. Do you believe the disinfectants we use to clean the barn between groups of pigs will cause the foaming by changing microbial activity?
I don't think so. I think normally if we were going to have a change in microbial activity there was a great paper by Zimmer probably 15 years ago now, and he showed that most of those antimicrobial agents tend to reduce microbial activity for short periods of time. I really think the reason that we get maybe more foam in between turns or when we normally would be cleaning a barn actually has more to do with there's no manure, there's no urine falling through, breaking those bubbles as we're doing it. There is also some work on anaerobic digesters that say you want to load them every day at the same organic loading rate, and if you do make sudden changes, you have a greater chance of foaming. So the microbial ecology, the propensity to make a product and decompose that product, get out of whack. Those are probably the two things that are causing it the most. Long term, you're right, some of those microbial additives could cause change in microbial community, but there's nothing I can say intelligently about how they would do that or what the impact would be, whether it causes just to move towards a foaming community or a non-foaming community.
Trying to read through all these yet. Okay, so when building new or remodeling, a lot of people are starting to remove pit fans and just use wall fans. How does this affect pit ventilation and foaming?
Yeah, I don't think it has a that big of effect, especially for methane. And you should really ask Brett Ramirez questions about this one instead of me. But most people who are removing their pit fans are putting their minimum ventilation fans somewhere in the barn. And what I really want is that ventilation, right? So methane is lighter than air. So when I tell you that pit fans don't, it doesn't need to be a pit fan, a minimum ventilation fan should be just as effective. It's working with this idea that most of that methane is staying near the manure surface for a very short period of time and then quickly diffusing into the barn. And as long as that's the case, it's going to be ventilated out, whether we're focused on pit fans or we're focused on above slat ventilation. Things like hydrogen sulfide, especially after pit agitation where it's heavier than air, there may be some small differences. Uh, Probably mostly small though, because if you think about a pit fan and trying to get air drawdown through the slats towards that pit fan, they tend to not be moving enough air to cause that to happen. So that's probably a minimal effect. But I think most of the research out there has shown that you need minimum ventilation. That's extremely important. But whether you're choosing to do it with a pit fan or choosing to do it with a sidewall fan somewhere in your barn, probably doesn't make it too big of impact on ventilation quality, but it might reduce emissions of ammonia and odors by minimizing what you have for pit fans and putting them on the sidewall instead of pulling more closely from the manure.
Another question here says we go from one extreme to the other with some of our barns. Some pits foam and some pits are crusted over. They use the same feed mill and same nutrition. What factors create this issue?
That's a great question. Okay, so we did a whole bunch of feeding trials as part of trying to understand what was happening here. And the good science I can tell you is that diet is important in microbial community, but it only explains about 35 to 40% of the microbial community that actually forms in a pit. The rest is related to something else, and that could be sort of water wastage in the barn. It could be temperature of the manure. It could be 100 other factors that I'm not prepared to tell you about. But what that really means is that 60 to 70% of the microbial community that forms is due to factors that we couldn't figure out while we were doing those experiments, right? It's just uncontrolled variability. So when one barn foams and one doesn't, you got unlucky, right? If I had two forests side by side, they won't develop the exact same forest. They'll be similar generally, but there's a chance they can diverge. And that's sort of what you're seeing there is that one diverged and said, there's not much potential to make methane, or I didn't trigger whatever it caused to get the right type of methanogen that processed that volatile fatty acid more quickly, and the other one did. So I know that's a terrible answer because it doesn't really help you out. But that's where we got, right? We couldn't explain that other 60 to 70% of variability that was driving what causes microbial community to form. It could be your pit additive. It could be how well you agitated and the amount of sludge you left. It could be that one was pumped before the other, and the one that you pumped first, you got more empty than the second. But I really can't say what all those other factors are. We were unable to identify them.
Perfect. I think on all the other questions, were either explained or kind of just said had the same question, just worded a little bit differently. Is there any final thoughts that you'd like to go over? We've got about 8 minutes left in the webinar.
Just really focus on safety, be vigilant, measuring if you have foam present. If there is foam present, be extra cautious. Even if there's no foam present and you're going to do hot work in your barn, be thoughtful and cautious. We should be keeping that material out of our pit to start with, but we definitely want to be avoiding fires. Certainly, I think we learned a lot about foaming, and I know some of those answers are unsatisfactory. There's a lot that we still don't know about manure. We'll keep working at it, trying to understand what some of those causes are. But hopefully you heard a few things today that might be helpful. If you need more information, I'm still in Denmark the rest of this week, but give me a call next week and I'll be happy to try and address them.
Perfect. Thank you so much. And I know that the Pork Industry Center is also working on putting out some information that's going to be geared more towards safety. So those will be available on our website as well. And any other information we get from Dan, we will make public as soon as we can. So thank you so much.